earlier. If I can find this. Yeah, so there was a question about whether we map data to other strains as well. And no, we do not, for the reasons that Omar described. But if I share my screen for just a moment, I'll show you here an example from another workshop, in this case for fungi. In this case, this was an analysis not of two different strains, but of two very closely related species, Coccidioides immitis and Possidacea, which were analyzed for both the free-living and parasitic forms of those fungi. And you can see that in non-strand specific data, where it's difficult to figure out what strand it ties to, we can certainly see that this is a gene that is upregulated in the parasitic stages of C. immitis. This is part of a cluster of genes that is present in both Coccidioides strains, but absent from the more distantly related Aspergillus nidulans. And when we turn to take a look at C. Possidacea, we can see that it is a gene that is also upregulated in the parasitic stages of C. Possidacea. So in this case, this is sort of what you were asking for, which is to be able to take a look at, to align the reads from two different related strains, or in this case, two different related species. But I've cheated here through the miracle of PowerPoint. This is not an analysis that you can do side by side in FungiDB. I've had to run those analyses manually and then cut and paste them and align them manually, because you will see that if we align these two genes don't align precisely. We can turn to take a look at another gene, for example. Here's another gene that is also inserted in Coccidioides relative to Aspergillus, that is dramatically upregulated in parasitic stages. And when we look at C. Possidacea, it is also upregulated in parasitic stages of C. Possidacea. But interestingly, you will notice there is no gene here in C. immitis. So you might be curious as to why that is. Maybe this is a gene that wasn't annotated because it is no longer functional, but in fact, it simply wasn't annotated because the annotation was poor. And if you dive into the methods from the paper that reported this work, you'll find that because they recognized that the annotation of C. immitis was poor, the annotation of C. Possidacea was poor, they simply transferred the annotation across from one species to another for their analysis. But we could now go in Apollo and add this gene where it quite clearly exists. So let me stop there and encourage you all to work through these exercises, including the Apollo annotation exercise if you wish, and that you run the query to identify genes with introns for which there is that are at odds with the annotation evidence for whatever species you may be interested in. We've checked this out for many different species, for Anopheles, for Ixodes, for Plasmodium, for Fusarium, Magnaportha, Cryptococcus, and it's quite informative for all of those.
So I do want to also demo one more thing when we were talking about annotation, which I think is quite useful. I do want to highlight there were some questions about whether the demo, the number of people have to leave exactly at 1. There were some questions about whether the demo for the upload your own data will be recorded, and the answer is yes, and we'll make them available. We don't have yet a step-by-step exercise for doing that. This is a new functionality that we've added recently, and we will, well, in its form we've added recently. We've had some other ways of doing that before, and so we will be developing that hopefully over the coming months. The one thing, David, I wanted to show, and I can do it quickly, and that is because you did mention user comments in the beginning, and you conflated that with Apollo, and I just wanted to make sure to clarify the difference. As you saw with Uli's nice demonstration, that you can go in and modify a gene model, and there's in fact a question in the chat as well about who can do these modifications and how do we trust that modification, and how is it curated and so forth. And it's a really good question. So for the time being, anyone can go in and as long as you have an account, you can go in and modify a gene model and finalize it and say this is a gene model. And it will appear the next day in the community annotation track. It's not going to make it to the official annotation until we have the resource to do so. That's an important aspect of it. But also, when that happens, we typically have ways to curate those annotations and make sure there aren't any, just weird changes being introduced and things that are affecting or actually taking things a step backwards. Uli, I'm sure you can comment on this as well, but at least in the past experiences where we've done this with communities where we've gotten a number of annotations from community, there weren't any really bizarre or crazy changes. We've managed to catch most of those. Is that accurate?
Yes, and we have automated checks as well. So yes, we do check the annotation.
Cool.
But that does not, that in any case does not replace the official, any information there, correct or not, even if it slips through the automated checks, do not replace the official annotation unless and until that's reviewed. It shows up in a separate track indicating community annotation.
So the other thing I wanted to show was that again, just to remind you that there are many searches that are available to you, and one feature that we have is a feature that allows you to add something called a user comment on any gene. So you can go to any gene page, and assuming you're logged in, you just can go in there and open up the user comment panel and add a user comment. And I will show you how you can do this quickly. But in addition to that, there is a search that allows you to return every single gene that has a user comment on it. And those can be potentially quite useful because these are people like you who had something to say about the gene. In many cases, it's not really going to change the official annotation, but it may be useful information. Like I tried to clone this gene and I couldn't clone it, right? That's kind of useful information. Or we've published this information about this gene. It's definitely a kinase, and we've also found that it's localized to the apicoplast, or whatever information you have from application. Or simply, I was just reading this paper, and they actually mentioned this gene as a gene that's involved in regulating transcription. Here's the link to the PubMed ID. And so you can go in and under annotation and curation, you can search for user comments. So I'm just going to look for this in ToxoDB. I'm using it as an example database. And so there are 3,080. I must have run this yesterday too, showing somebody. So there are 3,080 genes that have user comments on them. And you can see here that these comments are coming in. They span many different years and time spans. But I'm going to click on the first one. And when you go to the gene page, you will notice here it says user comment. There's one user comment. So you can view or add user comments. When you click on view, it takes you to the section of the gene page that includes this user comment. So you can see here this person, Yongzhen Jia from Beijing Friendship Hospital, added this comment on December 3rd. And it says that this is a large mitoribosomal protein. Let's take a look at the annotation. L1P family, yeah. So it's giving it a slightly modified annotation, and it's interesting. And if you click on this, you'll get more information about the comment itself. The nice thing about it is that you have the information of the person who added the comment, and it's an option for you to actually communicate with them and get more information. Let me reload the page. Okay. If you needed to add a comment, you can just click on add comment. The form is very straightforward. And the point of this is an uncurated way for people to add their own user comments. So I'm going to stop here. And David, I know you were going to do a quick wrap up, but kind of we've been doing a wrap up basically all afternoon. So I'm not sure how much else you want to add, but I'll leave it to you before I go into my demo.
Yeah, I mean, I think in light of the time, I think there is probably no need to do a detailed wrap up. We have spent all day working on the genome browser in one form or another, familiarizing yourself with the vast amount of information that is available in the genome browser to be able to turn on and configure and manipulate tracks to get views that you would like, to save that information so that you can use those views in looking at other genes on other days, or share them with other investigators. We focused on taking advantage of that information to accurately assess gene models. I've often given the example of a former graduate student in my lab who did an outstanding job on characterizing the biochemistry of the aminolevulinic acid dehydratase in toxoplasma, rate limiting step in heme biosynthesis, and who solved a beautiful crystal structure and did detailed enzymatic characterization on a protein that turns out does not exist, because the annotation predicted that it would be translated from the first ATG. And indeed, when that gene was expressed in E. coli, that is, the protein was made. But the predominant isoform in the parasite is actually translated from the fifth translation initiation codon, which required Bo to go back and redo the biochemistry and crystal structure on the proper protein. That's information that he couldn't have had in the pre-RNA-seq days, but now you do have that information, so you owe it to yourself to make sure that you are familiar with the experimental evidence underlying the approximation that annotators provide. Finally, we've shown a way to be able to input information yourself through the community annotations track, or as Omar highlighted at the end of the day, the ability to add other information, key literature, synonymous names, results of having knocked out a gene and finding no phenotype, all of which can be entered in as user comments. So we encourage you to take a look at the exercises, including the optional exercises, and contact us if you have any further questions. And I think that's probably sufficient for a summary for the day. Do
Great.
any
Thank you,
other
Dennis.
facilitators have anything else to add from today's summary?
So people are obviously welcome to, I know it's 1 o'clock and that's when the official end of the day is, but we did buffer in some time to do a demo of uploading some data. This is a feature that will expand hopefully in the coming months and include more types of data. But so if you need to leave, of course, don't worry. I'm going to record this session, or the session is being recorded. But if you stay on, that's wonderful as well. I did want to highlight that for tomorrow, just so you're aware that we will be focusing on a lot of the functional data that's available in our resources. So focusing on how to run searches for transcriptomic and proteomics data, doing Go enrichment, for example, looking at single cell data that as many of you know, is lots of people are doing single cell, and we're trying to integrate that in ways that are useful to you. And then we will talk about orthologs and trying to sort of understand function of genes by leveraging homology across organisms. So that's tomorrow, and so hopefully we will be able to see you tomorrow.